Welcome to the Lindamood-Bell radio podcast. We explore topics in learning and literacy. Hi, I'm Dave Hungerford for Lindamood-Bell, and today I'm talking with Dr. Jason Yeatman, Director of the Brain Development and Education Lab at the University of Washington. But first I want to let you know that Lindamood-Bell Learning Centers help students learn to their potential. We teach the skills that underlie reading, comprehension, and math. Our success with students starts with our unique learning ability evaluation. We identify the strengths and weaknesses that affect school performance, and we develop individualized plans to improve learning. A school principal once described our learning ability evaluation saying, "It's like getting a fingerprint of your child's unique learning profile." This extraordinary evaluation is available at any of our locations or online. Find out more at lindamoodbell.com. or call us at 800-300-1818. Since this episode with Dr. Yeatman was recorded, some of the research discussed was published in the scientific journal Nature Communications. The article is titled "Rapid and Widespread White Matter Plasticity During an Intensive Reading Intervention." Get a link to the full article on our blog at lindamoodbell.com. I'm happy to be here with Dr. Jason Yeatman, an assistant professor at the Institute for Learning and Brain Sciences, University of Washington. Welcome.
Thanks for having me.
Dr. Yeatman, I'm interested in finding out your journey to your research and what has kind of gotten you interested in dyslexia and the research that goes along with that.
Well, first of all, as a cognitive neuroscientist, I think that reading is one of the most interesting things the brain does. First of all, it requires integrating many different types of brain circuits. So brain circuits for vision, brain circuits for hearing, brain circuits for language. That the process of even recognizing a single word requires that these highly disparate circuits communicate in an interesting and new and very precise way. So understanding how the brain's able to do that is just a fascinating scientific investigation. So that's what originally got me interested in studying reading. But the thing that's kept me fascinated with this topic is also how important it is in terms of how it impacts children's school, children's learning. That I truly believe that the information that we can discover as scientists investigating the basic mechanisms of reading in the brain can inform how teachers or special ed instructors actually work with children and teach them how to read, or how they intervene for children that are struggling.
And what are some of the methods that you use to measure and look at those abilities of reading?
Yeah, so of course everything starts with behavior. We're interested in real, live, developing children. So we first want to characterize, their reading skills, how quickly they can read, how accurately. But then the bulk of our research dives into the brain circuits that implement this cognitive function. So one of the methods that my lab's been a big part of developing is called diffusion MRI. So MRI, or magnetic resonance imaging, is the same machine that is used for clinical assessments of the brain, but also for any other, the knee, the back, the stomach. People go in for MRIs for many different reasons, and we can make this machine measure specific properties of the brain, such as water diffusion. And water diffusion provides a window into how the brain is structured on a cellular level, because water molecules are constantly moving around, bumping into the structure of the brain, allowing us to make inferences about how densely packed cells are, how coherently organized they are. So this method of diffusion MRI is one that we've worked a lot with in terms of developing specific ways to measure the neural connections, the bundles of wires that connect together this circuit for reading, and make inferences about the integrity of these wires, how densely packed they are, how quickly they can send signals.
So this is not measuring the blood flow like an fMRI would do.
Correct.
This is something different.
Correct. Yeah, fMRI, or functional magnetic resonance imaging, measures changes in blood flow that are caused by neural activity. This allows us to map the circuits of the cortex that respond to different types of stimuli, that, for example, become active when you read a word. What we're particularly interested in is how these regions of the cortex are actually connected together, the bundles of wires, the cabling that allows signals to go, say, from the visual cortex, where visual information is processed in the brain, to a language cortex where we produce and comprehend language information.
So how does the MRI machine actually measure that? To my knowledge, I think that the blood flow is tracks the iron in the blood, the metal. Is that right? How do you track the water in the MRI?
Oh, that's a longer physics lecture. I think you have to come to one of my classes for that one.
I got to enroll in the W, huh? So tell me about some of the particular studies you've maybe conducted in the past couple of years, readers. For example, let me just ask about one that I saw, the one with the motion perception and reading.
Yeah, so one of our behavioral studies of reading, there's long been a theory that reading difficulties are actually caused by a deficit in a particular piece of the brain that processes visual motion. And actually just that idea on its own is kind of fascinating. People take it for granted that your ability to see that something is moving, for example, track a moving ball and estimate where it's going to be so you can catch it, depends on a particular part of the brain that processes moving stimuli. And there's long been an observation that part of the brain functions slightly differently in people with dyslexia. And that's caused a long debate of whether this deficit in motion processing somehow causes disabilities with reading, or alternatively, maybe it's somehow caused by differences in people's reading skills. And we conducted a study where we looked at motion processing abilities in a group of subjects that are actually going through the Lindamood-Bell intervention. So we looked at, first of all, their improvements in reading scores over time, and whether there was a relationship between their abilities to track visual motion and their development of reading skills. And we found that these visual motion processing abilities, they were correlated with reading scores, but there didn't appear to be a causal relationship where deficits in motion perception actually cause children to learn in a different way.
How do you measure the motion perception?
To measure people's motion perception, we develop some experiments that have people track moving dots on a computer screen, basically measure how accurately they're able to track the movement of these dots. And some people, it's very easy. They're able to very accurately see the direction and speed that these dots are moving. And other people, particularly those with dyslexia, struggle with this kind of simple motion task.
So one would think that, like a baseball player that has really good maybe eye, hand, and predictive ability of something like a ball, there should be no dyslexic baseball players.
Yeah, that's what one might think. I don't know if it's true or not, though. So, as I was saying before, one of our focuses has been on the wiring of the brain. And first of all, looking at how the reading circuitry is wired together, both in the typical brain and also in the brains of children and adults that struggle with reading. So the first series of studies just mapped out how is this circuit connected together? What's the trajectory of these cables that allow information to flow between the reading regions of the brain? And next, once we identified the structure of this circuit, how it's typically organized, we ran a series of studies looking at how that differs in children who are particularly strong or particularly weak readers. And we actually tracked developmental differences in how these physical connections in the brain develop in children who are relatively strong versus relatively weak readers. This has been a big part of developing a theory that dyslexia actually emerges from differences in the connections between brain regions.
And that fits in with the white matter and the cable processing you've been talking about today. I should say you just finished a long talk to Lindamood-Bell staff on a current, it's in the publication process.
Mm-hmm. Yeah, we've been doing a long collaboration with Lindamood-Bell. And as I said, that there's differences in how these cables, how these connections in the brain develop in better versus worse readers. And what we're interested in is what happens when we intervene, when we take someone who's a struggling reader and we put them through an intensive intervention, say with Lindamood-Bell. Does that actually change the physical structure of how these brain regions are connected together? And I guess you'll have to stay tuned to see how that turns out. We're still finishing up that research project as we speak.
Great. Well, we'll look forward to that. You're not going to give up the ghost on any of that, I guess. Huh?
Can't give up the details until it makes it through the peer review process.
What other projects do you have kind of in the hopper?
Yeah, a big one. So we're also interested in how we can capitalize on technology to help children and adults with reading. And we have another big project that's aimed at measuring differences both in how the brain is structured and also how different perceptual systems for vision or hearing function, and trying to develop targeted technological interventions to help people based on their kind of unique profile of how these systems function. These can be simple changes such as how fonts are rendered on a computer screen. That we've actually been finding that for some people with dyslexia, if we change the spacing between letters in a word, it actually makes them read slightly better. It's not a profound change. It's not going to take a dyslexic child and make them a superstar reader just by changing the font. But we have found that we can reliably detect differences in how some people with dyslexia see the world and subtly change the spacing between letters and font to personalize it specifically for their unique visual system.
I just saw an article yesterday online about somebody who had changed the typeface completely, had like taken the tails of the Y and actually kind of shifted the direction so that, for example, B's and D's were drastically different, and the stems coming up on them were, one was wide at the bottom, I think the B, and D was wider at the top, and so it made a real distinct difference. And the intent was to help dyslexics with that.
Yeah, and I think that the big goal of this, I see having more ties between scientific research done in an academic setting and real applications, be them corporate applications or not-for-profit applications. Having more deeper ties between research and practice is really important thing. There's a lot of great ideas in how we can change fonts or how we can leverage computer displays or text-to-speech technologies to help people with different types of reading skills. And I think there's a lot of room to make progress there. And the important thing is going to be to tie technology development to solid scientific research to make sure that products that are developed and sold to people have a good sound research backing, and that the research can inform how to optimize and personalize these products.
You have Microsoft there. Is that part of your job in your lab to develop partnerships with outside sources outside the university?
It's not part of the job per se. Some people in academia think that it's important to have kind of barriers between corporations and academic research. I feel differently. I think it's a really strong opportunities for collaboration when there's mutual goals. I have a wonderful collaboration with the Microsoft Advanced Reading Technologies group. They're the group at Microsoft that's developing fonts, that's developing different applications to support reading. And we've been talking with them for years over kind of joint ideas over how we can leverage basic scientific findings in people with dyslexia or people who read in different ways, and use that to inform how they develop products to help these people. So for me, that's been both scientifically fruitful and also hopefully doing something that translates to society in a nice way.
Have you looked at, talked about reading the typeface on computers, the effect of the screens that most of us are, and certainly in those developing years for kids, have a phone in front of them, they've got a computer in front of them, they've got a tablet in front of them so much of the day. Have you looked at or have any gut feeling on that?
I haven't myself. I do have gut feelings, but whenever I'm sitting on a podcast or a interview as a scientist, I try to stick away from giving my gut feelings on things, because I always think that our job as scientists is to take evidence that we collect in the lab and write it up in papers, also present it to people in an unbiased way. I have many opinions on many things that extend far beyond what I've discovered in my lab, and I try to only share those in a private setting, opposed to a setting that carries my name as a scientist behind it.
Fair enough.
So in the goal of creating deeper links between academic science and society, one of the projects my lab's undertaken is developing a series of learning modules to distill the information in the scientific literature on reading and dyslexia into an accessible format for parents, for teachers, for children, for people that are interested in learning kind of the current state-of-the-art in science on literacy. So on my lab webpage, you can find some links to some online learning modules that we've developed. And this is work with a graduate student in my lab and also with the whole outreach team at the Institute for Learning and Brain Sciences, where my lab's located, to build these modules to try to translate what might seem like difficult and complicated and convoluted scientific findings in the large scientific literature into hopefully something that's more digestible for people that want to inform themselves on the science.
And what is that website?
So my lab website is braineducation.com, and from there you can find links to some of these learning modules we've created, to PDFs of our published papers, and also in some cases links to talks I've given on reading, dyslexia, and brain development.
Can you describe your lab and what the different work and areas that are a prime focus for you all?
Yeah, my lab is called the Brain Development and Education Lab. We're located in the Institute for Learning and Brain Sciences, which is an institute for interdisciplinary research, and as the name suggests, focused on learning and the brain. It's at the University of Washington. And my lab is an interdisciplinary group of psychologists, neuroscientists, educators, speech and hearing scientists, engineers, computer scientists, and we focus on understanding the neurobiological basis of learning, and particularly the process of learning to read. And we take an interdisciplinary approach to this, where parts of it is developing software for analyzing data and fitting statistical models. Other parts of it are designing experiments to probe reading in different ways. And then, of course, another part of it is conducting brain imaging experiments where we look at the brains of children and adults with various reading skills.
And are you in the classroom yourself teaching?
I do, and I also teach. I will be teaching a class called Speech, Language, Hearing, and the Brain as soon as I return back to UW.
Nice. Well, thank you so much for spending time with us, and wish you well. And
really a pleasure to be here. Thanks, Dave.
thank you for listening to the Lindamood-Bell radio podcast. I hope you listen to more episodes, and if you enjoyed this, please subscribe. Follow Lindamood-Bell on Instagram, Facebook, and Twitter @Lindamood-Bell.